a pioneer center in the Middle East with a number of well-known and prestigious professors in studying human genetic disorder, especially the Mendelian disorders. Dr. Rabab Khairat was selected by the National Research Center and Supreme Council of Antiquities to be part of the first Egyptian team to discover more about the genetic makeup of the Egyptian mummies, especially the royal mummies in the 18th dynasty. Therefore, she was a member in the research team that founded an ancient DNA lab in the Egyptian Museum with cooperation with Discovery Channel between the years 2006 to 2009. Since then, she's a part-time member of a team in Ancient DNA Lab, Egyptian Museum, and a member in Paleobiology Committee in the Egyptian Supreme Council of Antiquities. In 2009, she received a DED scholarship in Institute of Human Genetics, Division of Molecular Genetics, University of Tübingen in Germany, where she had finished her PhD degree in biology and learned more about the next generation sequencing technology, or rather NGS. Just to highlight to the attendees and to everybody who's listening to this talk at the moment, please write your questions down and keep them until Dr. Rabab finishes her talk. You can then at the end raise your hand or put on your mic or tap them up, and we will ask Dr. Rabab to answer your questions. So I will ask everyone who's not going to be speaking, can you please switch off your mics, or rather mute them, mute your mics. Only Dr. Rabab is going to have her mic on, and only at the end you will put on your mics when you want to ask questions. Welcome to Dr. Rabab Khairat, and over to you, Doctor.
Oh, thank you very much, Pablo, for your presentation of me, and now, sorry, I lost my slides. Yeah, now it is come back. Sorry again for this problem. Thank you very much for your presentation. I hope you will like our talk, and I hope that it will be just interesting for all of you. Let me go to our next slide. So since a long time, the scientists used the fossils and the bones to reconstruct and come up with theory about the revolution and how it is moving. And then after a while come up the DNA to the scientific community, and a lot of scientists start to study the DNA from the modern sample, as well as some also were interested to study also DNA from an ancient sample. So what can be the sources for the ancient DNA samples? We can just give here a few examples like mummies, plant materials, organisms which can be also in the ancient samples. So this can be the source of the ancient samples. What can be interesting for studying the Egyptian mummy? I will be interested to study more about the mummy's history, identities, to know which one is which one and which is belong to this family or to this dynasty, to know more about the mummification secrets, to know more about the ancient Egyptian population and the common disease at this time and its pathology. All of this was so interesting for a number of scientists, and to just give you more information about what happened to the DNA after the death. It usually or typically decays with the body. So decays by the nucleases, by microbial decompositions. Usually there is a number of sites in the DNA which can be easily affected by the decay process. Can be depurinations like loss of purine base, oxidative damage, hydrolytic damage. And what can be stop this decay can be stopped by rapid dissection or low temperature or being in high saline solution. However, with all of this also should be a matter of decay, but it will be more slow than what is just poured in the soil. The first studies was in 1984 with a group studying guaga. This is a form of zebra, and also it was interesting for Professor Pabo to study an Egyptian mummy also in 1950, in 1995. And at this time started the revolution of the PCR and using it to get a lot from the same DNA sample. Because the cloning at this time, using the cloning after the DNA extraction causes a lot of problems. So and with the revolution of PCR, starting a new protocol for studying the ancient DNA. So use the PCR, make it possible to not clone the DNA directly, but clone it from the PCR afterwards. And for sure, this gave a chance for the scientists to study a specific target of the genome. And for this, most of the studies from this time to nearly 2000 used the mitochondrial DNA as a typical target for ancient DNA study, because the mitochondrial DNA is in high in relevance to the nuclear DNA, which make it more easier to catch it by in the so old sample. And for, I don't know if you are familiar with PCR or not, the PCR used a specific primer, which can be specific for a specific part of the DNA, and then it is multiplied by the thermal protocol, and then this PCR product, once it is visible in gel, it can be sequenced by Sanger sequencing or sequencing machine. This is just the Sanger sequencing machine, and here it is what is inside the machine. It is shortly it is rely on the theory that is we used a specific dinucleotide base pair with a specific fluorescent, and once it is with the template, there is just like a T nucleotide, it put T and give a color which is captured by the detector, and it can be saved with the computer. And then you can read the sequence of the DNA with these peaks, and you can differentiate by your sequence by GAT according to the fluorescent signals which is detected by the laser. And for decades, this is what the way that is the people dealing with the ancient DNA. That is, they use the PCR to amplify a specific part of the DNA and sequence it. And then to be sure about what is there in this sequences product, because it can be different product in the same vial of PCR. We used to make a cloning from all this PCR samples, PCR product, and see what is the major sequences in this product. And once it is the major, we use it as the authentic PCR sequence. And since then, the committee and the scientific community used a specific criteria to make it like a guidelines for ancient DNA studies. Like that, we have to make biochemical assay for macromolecule in the same sample to be sure that is this sample, or to give us a clue about the preservation of this specimen. Like to have a different places for extraction DNA from this place of the PCR to avoid a contamination of the DNA samples by the PCR's product, since it is more abundant than the DNA in the ancient sample. Also to use just a short PCR fragment, not a long, since it should be the DNA samples, ancient DNA samples should be fragmented, so it is more logic to have a short fragment from the long fragment. And also this is like a selection of what should be an authentic DNA more than the long fragment. Also to have quantification of the PCR. And we have to be sure we can see it is good PCR once it can be visible on the gel in different 3 PCR reactions. Also, they also from this guideline to exclude some of the mitochondrial sequences, which is in the nuclear sequences or the nuclear genome, to avoid this from coming from the nuclear, because also it will not be an authentic mitochondrial DNA. It can be also authentic nuclear ancient DNA, but it is not authentic nuclear mitochondrial DNA. Also, it should be amplified in a second place, a second away place, from independent extract. Reproduction of the second, which is called like a reproduction in a second laboratory. That it should be the same result done in another independent labs with an independent extract and come up with the same result. This guideline burden the scientists over decades to work with the ancient DNA samples. And since then, there is a lot of studies come up studying different pathogens, like Dick and Nerlich in 2003 and Nerlich in 2008. Also, those which concerned with relationship conformation, like those was done with Romanov family in 1994, and also in 2009. And also in our publication done with the Khairat family in 2010. In 2005, start a new decade. This will be the second part of our presentation. But what was the problems with the PCR technology? The problem with the PCR technology, as you can get from my guideline, what I present in the guidelines or the last few slides, it is took a lot of time to come up with what can be an authentic result or a confirmed result to present to the scientific community. Also, it was so hard to come out from the samples with all of this DNA for all of this scientific steps. So the paucity of our sample, the troubles with the traditional technology, PCRs and Sanger, troubles in comparison with output, the effect of the resin or whatever the other coming by extract in the DNA, which inhibit the PCR reaction. All of this make it like so hard to get to work with the ancient DNA samples with this protocol. And also with the PCR, there is an advantage to amplify the contamination DNA from whatever the sources in comparison with the ancient DNA, because there is a step of a high amplification done in the PCR, which can be for sure amplify what is much redundant in the sample in comparison with the ancient DNA. As I told you, in 2004-2005, started a new decade in the scientific community. Started with the next generation sequencing technology. Once it is available to in the market. What is the NGS technology? The NGS technology, it is a technology giving the scientist the ability to sequence a lot of genetic material all together, sequence them all together in a short time with a specific protocol, starting by fragmentation of the DNA to small fragment, and then just repair the end, because mostly this fragment will not be applied in, and then ligate it with a specific this end will ligate it to a specific adapter to be used with a specific primer to amplify it shortly, not that much, and then used for the sequencing by the machine. For the NGS, you can also have the probability to have a much more than one barcode, like a detector at the start, so you can differentiate between different samples can be added together in the same run of the NGS. So you can use different samples with a different barcode, and easily afterward by the analysis step, you can just group those samples with specific groups with specific barcodes and use it separately from the others. What is the available NGS technologies in the market? There is a number, and this is where the most probable and the most famous platform in the market, like 454, the Illumina, which is the most probable and most common in the market and in the different labs now, since it is quite good with the price and the quality and so on, and also the solid platform and the ion torrent platform. Whatever the platform used is the same protocol, using the same DNA sample, cut it to fragment, ligated with a specific adapter, and then make a step of amplification shortly, and then used for the sequencing. The sequencing used to be like whatever the chemistry of the platform, it is like that you used a compartment or a slide, you put a specific adapters to amplify again this fragment of DNA separately in this slide, and whatever the chemistry used for in the platform, there is a captures of the sequence of the nucleotide, the nucleotide in the sequence, and this image processing used afterward by the supercomputer to be translated to a DNA sequence. So this is just a technical part which will be difficult for all the people to go in. So whatever is the chemistry, the same protocol is done outside the the platform, and the result come out also is quite similar with just a few changes. For sure, there is a difference between those platforms, and there is a positive point and negative point, which make it for all the people and for all the scientists, like they have to to choose according to their positive or negative or their application and their requirement, because like for the long fragment most of the people used 454, so this will be useful for in the assembly of new genome. For the high result, and perfect result, the people like in using solid for the definition of new SNPs. And for the most common labs, the people used Illumina because the error rate is quite good and the fragment reads is quite good, and it is used for many applications. However, it also had some problems with a specific part of the genomes, but still it is quite good for most of the application. So it is a matter of equilibrium. That's the scientist can choose the platform according to their requirement and their application. Since 2004 till now, there is a lot of effort to improve the technology, to reduce the times of the sequencing, and to increase the read length, to decrease the error rate, because all of these platforms still have a problem with error can be done by their chemistry. Also to develop a bioinformatics tool to make it more easier, to make it more available for all kind of scientists to work with, to reduce the runtime and the cost, and for sure the driver for all of this was the change and the improvement of the computer science. Just shortly, I would like to tell you that also there is now the third generation sequencing technology, which is the difference between the third and the second. That is the PCR, the step of the amplification in between the ligation step, and the sequencing is not present anymore. And the sequencing or reading the base pair of the DNA fragment is done within the time of the amplification. So it is just one step amplification and done with the sequencing, which make it much lovely for all the people, for all the scientists, because then we can exclude this error rate from our data. However, still it is developing, so it is not that perfect yet, but it is a new hope for all of us. Since 2004 or 2005, the ancient DNA studies take the advantage also from this NGS technology, and especially that is the ancient DNA is already should be fragmented, so we can just exclude the step of fragmentation and use the DNA as it is for the NGS library preparation. And this will be also give the chance for the ancient DNA sample to be more redundant than any contaminant DNA be around in the sun. And so this is just a few examples of the studies done with the ancient DNA samples since then. The cave bears study, Mammoth, Neanderthal, Iceman, and also our study which is done with the Egyptian mummy. What is done in what was the aim of our study? The aim of our study was to determine the degree of information can be gained from the mummified samples once it is analyzed by NGS. And our aim or our idea that we will make like sequencing for a number of Egyptian mummies to see what is there in the sample and compare them with another samples from whatever the available datasets in the internet, or from skeletons which we had it from lowland samples. And so that's we used 5 samples from Egyptian mummified heads in between the 3rd intermediate period to the Roman period, and 2 long bone from a lowland Bolivian skeletons. And this was our samples which we used in our study. And then after we come out with the datasets, we used other datasets from literature like Saqqaq, Iceman, and Denisova for the comparison with our datasets. Just few terms. I don't know if you are familiar with the NGS terms. What is coverage? Because we will use these terms in presenting our result. Coverage, which means that's what are the numbers of time that specific region of genome can be covered by the sequencing reads or sequenced read. So if we said it is 10x, mean that this reads cover 10 times by the sequencing reads. And what is the unique reads? Mean it is just one each read covered 10 times by unique reads, mean that it is not from multiplications by the PCR, because we had in the analysis we can exclude those done by multiplications of PCR, and we just use those unique reads to be sure that it is not an artifact from the PCR amplification. So that's why we use just the unique reads to say that it is covered by 10 times by unique reads. This means that's covered by different reads cover this region, and it is not come out from the PCR replicates. The complexity, it is the percentage of the unique reads in our data, so or datasets, the resulted datasets. So this is the term which I will use in presenting my my result. So from our datasets, one of the Egyptian mummy, for sure. This is not the perfect result, but this is because the samples which we had at this time was limited, and also it is still, some of them are tissue, some of them are bones, and it is a little bit a small sample.
Hi.
As I told you, there's inadequate numbers of reads was mapped to the mitochondrial DNA. There are about 3.5 reads. From them, it is like 31,000 reads, unique reads, was mapped to the mitochondrial DNA, which can be, in another word, be translated to that's the mitochondrial DNA genome was covered in average by 190x. So this is what I finalized in my dissertation's PhD in 2013, and just to be to know more about the pipelines of the analysis of NGS datasets, the datasets come out from the NGS machine in a format of FASTA-Q file. And this FASTA-Q file, we then exclude or remove the index which used for the sequencing, and we make like a quality control of filtration of the reads, and those which is just fine with the quality we used for mapping against the reference, which is, in our case, we used the human reference. And then we convert it to a specific format file which is called SAM, and then we use the BAM file from the SAM, and we use a viewing software to view our sample, our data in our ancient DNA samples. And once we are happy with our data, we use it another software to detect different SNP and make a filtration of it. This is the common pipeline of the NGS datasets. And this is the what come out from our sample. It is a snapshot of a BAM file of the dataset from the Egyptian mummified sample. The coverage of our reads of the mitochondrial DNA was, as I told you, 190x using the unique reads. And this is just like a snapshot of 390 base pair. And you can see here, this is this was at the start of the mitochondrial DNA genome. And from this part you can see that the coverage sometimes go so high and sometimes go down, but in average it was like 100x or 190. And this is colors. It is the difference between our result and with the reference mitochondrial reference genome. So from using other software, we could grab those information of the difference between our result and the reference genome, and we can use it for SNP detections. And using a software which is called VARScan, we could detect the haplogroup of those of these samples. So we used a specific SNP in the hypervariable regions to detect that our sample is belong to I2 haplogroup. And when we search for this haplogroup, it is one of the so rare haplogroup over the world. It is just scattered in specific groups somewhere here and there. And our theory that it can be also be in our population in all time, and it vanished by time through the movement through Egypt to the other places in the world, because it is was scattered in a specific group in so high in so north in Europe, and in other group in the Middle East, and also in specific groups in Arab Peninsula. So so we saw that it may be one of so old haplogroup, and it is vanished by time. So it is just like insight, as you said, that it is just the first insights of the genetic makeup of an Egyptian mummy. So now there is another part of our talk, that as we deal with our sample, since it is a human sample, so we used it and we mapped it with human reference, and we use this human reads for detecting more or know more about the human genetic makeup. But now the samples of the ancient DNA can be considered like metagenomic sample. What is a metagenomic sample? Meta is mean that's different materials together. Genomics mean it is different genomic material together, because the ancient DNA sample is used to be an example of different genome together. Microorganism which is left over this body over years, the microorganism which was around this body over years, the plant around the ancient DNA materials or body, maybe also some information or genetic material from the mummified materials, the mummified embalming material used for the mummifications of the mummy. So that's why since 2004 also most of the studies dealing with ancient DNA, dealing what is there with the ancient or the original DNA in the ancient DNA sample. That means that we studying a human bone, so we dealing with a human genetic material as well as with other microorganism or plant or whatever genetic information in the same sample. So how we can make this? At this time we use a different pipeline of the analysis. We use the same pipeline like the common with mapping with a human reference, but after the mapping with human reference or whatever the reference, we make a filtration of this mapped read to the reference and use the rest to BLAST with the in our nucleotide database. And this nucleotide database is like a mix of all the available genetic information till now, and is used to be updated over and over time. And we use this information to see what is in our sequencing result can be mapped to other microorganisms, others plant, others parasites, and we use a different software which is called MegaBLAST to view this result. So from our result, this is just a snapshot of this MegaBLAST software. We found a lot of reads mapped to specific types of trees or genus of trees, pinus, and this pinus over and over we found it in most of the literature as a used tree for the gum or come out from this tree used for the embalming and the mummification of the Egyptian mummies. As well as we found also a numbers of reads mapped to a numbers of parasites like Plasmodium, Falciparum, and Toxoplasma. And most of those information cannot come out from no source. No, it should be something around or something in our samples which make it most prominent in the rest of the reads come out from our sample. So that's why we used a specific primer for this pinus. As example, we used a specific primer for a specific species of the pinus, and we successfully got a PCR fragment from our sample, and we confirmed it by Sanger sequencing. As well as we done this also with Plasmodium falciparum, and it is also published in another publication. Just shortly, we make this comparison between our samples or plus the Bolivian skeleton, which we called the warmer climate sample group, and also with the available datasets in the internet or the literature from Denisova, Saqqaq, and Iceman, and we come out with an interesting finding that's most of the Egyptian mummies, and the Iceman had a specific high percentage of most of the Egyptian mummy had a high percentage of firmicutes, which is bacteria lived in a so hard environment, and which is also so logic that's to find such an amount of firmicutes with such an hard condition like this, because most of the mummified tissues have a high concentration of salt. As well as we find that also the Egyptian mummies and the Iceman had a high low concentration of actinomycetes, which is the soil bacteria, in comparison with the other samples come out from the soil, like the Bolivian skeletons or the Denisova or Saqqaq, has a high concentration or high reads mapped to the actinomycetes, which make it like this easy to group the samples come out from mummified samples, since it is used to be compared in a specific coffin or somewhere away from the soil. From those come out from the soil. And finally, we can say, and what we can come out from our study, this is NGS can be a powerful technology to get more information from the Egyptian mummies or whatever the source of the mummies, and it can give a great push to the molecular Egyptology. And that is the Egyptian mummy, like all the other ancient DNA sample, can be a source of genetic information, and it is a matter of preservation status. And this is the human DNA content can reach a sufficient amount once the samples is good preserved, and it can be like an interesting and a way to know more about the embalming materials, and to know more about what is can be like a unique signature for mummified sample. And thank you very much for your attendance. I hope you like it.
Thank you so much, Dr. Rabab, for an interesting talk. And for me in particular, I learned a lot as someone who is new to this area of research. So thank you so much for explaining your presentation in such an interesting way. It is now time for questions. And in the interest of time, please keep your questions nice, sweet, and short so that we get more people asking questions. We will take 2 questions at a time, and I will ask Dr. Rabab to respond. So we only have 12 minutes at the moment for questions and answers, so please keep it short. Who has you raise your hand, I will accept. Samar has a question. Approved. Ask your question, Samar. Samar, you can go ahead and ask your question. Samar?
Yes, I'm here. I'm available for any kind of questions.
Go ahead. Okay, Samar says it seems as though her voice is not working. I've given you access. You are not muted. It might be the settings on your computer, and that might take time to sort out. So in the interest of time, can you please tap down your question? I'll read them up, or thank you so much. As Samar is typing a question. Could we please get another question either typed or someone raise your hand, and you can also ask your question as we're waiting for Samar to complete typing up a question. I'm reading Samar's question, and I'm going to ask that anyone else who has a question, can you please ask it? This is what Samar Kamal Kasim has said. So she says, so you have results from plants and other bacterial genomes. What kind of data did you find in the human genome itself? I repeat, what kind of data did you find in the human genome itself? Thank you, Dr. Rabab. You may answer the question.
It's okay. So as I present in my presentation, we got enough information from the mitochondrial genome. And to get information or clue about the haplogroup, this is quite good to know from the mitochondrial genome. So this is the first information that you ask about. The data which I come out from the human genome, it is the haplogroup of the mitochondrial genome. And this is like the first try with Egyptian mummies. So for all the ancient DNA studies done by the NGS, they used to make like a screening study to use it for choosing a specific sample for further investigation. So to find like this information that you find that you can get more about the bacterial genetic materials that decay the mummified tissue, to know more about the plant around, to know more about the human genome, that you can get data from human genome, and to get information from about the mitochondrial haplogroup. This is quite enough information.
Okay, sure. That's interesting. Samar continues with another question. He says, did you map these findings on or to the recent Egyptian genomic data? Did you map these findings to the recent Egyptian genomic data? Can you please respond to that?
Yes, sure. Actually, unfortunately, there is no Egyptian genomic data so far. The NGS technology is starting since 5 years or so, starting to be used in Egypt. So we are a little bit a step behind the what is available over the world. So, but we had such the aim to make our there is a numbers of running the project, and we collaborating in doing some also. And once there is available Egyptian genomic data, we will use it for the map. And also we are planning to use NGS technology with a numbers of Egyptian mummies. This is our also running the project now. So this is, as I told, it is just a start, but this study is taking time to be available for scientific community.
Okay, sure. Thank you so much. That is very interesting. Again, people are typing questions. You can also raise your hand on the top bar of the computer. There's a mini poppy person who's raising their hand. You can press that and raise your hand. I'll continue reading the questions on the side. Dr. Rabab, can you answer the following question? Aiton Menkis says, very interesting, thank you. He continues to ask, does finding P. falciparum, I don't know if I'm pronouncing that right, falciparum, suggest that the individual suffered or died of malaria, or are there other explanations for finding PF like mosquitoes themselves? Dr. Rabab?
No, it should be. No, because we didn't use the mosquito for our study, but I guess it can be, yeah, once we find these sequences, because we use a specific primer, so specific primer for PF. So once we got this information, genetic information, it should be that this guy had malaria. So if he died by it or he survived by it until his death, we don't have this information. But once we got this information from his phone, this means that he had PF. And this was suggested not by only us, but Dr. Nerlich and Dr. Zeng, I don't know when exactly, had a numbers of publication confirm the presence of our infection by Plasmodium falciparum by the presence of such genetic material in ancient DNA sample. So it should be a confirmation that this body infected by malaria.
Thank you, Dr. Rabab. Now there's a question from Dr. Faisal, who asks, from which part of the mummies did you obtain a sample for DNA extraction? Can you please answer that, Dr. Rabab?
Yeah. Yes, sure. I did this work in Germany with a specific collection in Tübingen Museum, prehistory museum. At this time it was not there even, but it was just a skull. So we obtained what was available for us. We had like from the back of the neck, we had tissue samples, and also we had samples from the vertebra back in the head, back in the neck. So this was the available sample at this time. But for sure, we are planning to get more samples from other parts of the body and use it for NGS studies. And this is already mentioned in my publication also.
Okay. Thank you, Doctor. We have 4 minutes remaining for the presentation. So anyone who has any last questions, can you please raise them now, or I will give Dr. Rabab to say a last few words. I see that Emmanuel and Marina are typing. I'll give you a few seconds. Okay, so there's a question from Emmanuel, which asks, which challenges did you face? It's multiple questions. Which challenges did you face when carrying out this kind of study? Secondly, what recommendation would you make to anyone interested in doing an NGS study in Egypt or any other African country? Can you please address those, Dr.
Actually, the challenges with the ancient DNA samples is much more than any other samples. So it is not easy to dealing with it, because there is a lot of inhibiting material come out with the DNA extract. Make it is not that easy to go further with library preparations steps, and so we make a lot of modification to the known protocol to come out with an adequate amount of to come out with adequate amount of reads. So we make our own troubleshooting for the protocol to come out with this information. So this is the first challenge, to work with the ancient DNA sample itself, and also with the Egyptian mummies, because the resin which is used for the embalming, for the mummification, is also not one of the so strong inhibiting material. But we find our way to work with such a kind of samples, and for sure the dealing with NGS technology, it is not that cheap technology, and all the running materials and supplies is expensive. So make it also challenging, but more or less it has become more cheaper now. And just not forget a point, he said the challenges using the NGS or this kind of study. And in Egypt, as I told you, that we are since 4 years or 5 years we're starting using NGS technology in Egypt, and even there is not that high throughput, high technology. We had just a few benchtop NGS technology, and this is make it also not easy to dealing with the NGS in Egypt. But I hope that so far recently we will have the ability to through others company, because nowadays there is a surface in Illumina company and also in Iron Torrent company to make the library or whatever in your lab, and you use their facilities for sequencing, and this is make it more easier to dealing with NGS technology in Egypt.
Thank you so much, Dr. Rabab. We just reached 4 p.m. at the moment, so I'm just going to say, in the interest of time, that we stop at the moment. If anyone has any more questions, I'm sure there's many more questions, as I haven't even asked 2 more that I see on the chat. Could you please email Dr. Rabab Khairat on her email, rababkhairat@gmail.com. The email, I just put it up there at the moment, so continue engaging with her. Dr. Rabab, thank you so much for such an informative and interesting talk on the first insight into the Egyptian mummies using NGS. We really appreciate you taking time out of your busy schedule to give this very informative talk. We really appreciate it. To everyone, thank you for attending. We're going to have another webinar with a different topic, not in July, but in August. And we'll send out an email through MailChimp, and we'll promote it on Facebook and other social media platforms. Thank you so much for attending, and have a good day, everyone. Goodbye.
Yes, bye.